Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.
So hi, Dr. Palomero-Gallagher. Thank you so much for your time and participating in this interview today. We would love to learn more about your journey in brain mapping and your upcoming OHBM keynote at the 2024 conference in Seoul, South Korea. So our first question is, can you please tell us briefly about your journey in brain mapping?
Right. Well, I've always liked looking down a microscope, right from when I was in school. So it's sort of like natural that I moved that way, and I did my PhD in Düsseldorf University. Also, I studied biology, but I ended up doing my PhD at in the medical faculty, Düsseldorf University, and it did involve using the microscope, but I wasn't just looking at the cytoarchitecture of the brain. I was also comparing it with the distribution of receptors for classical neurotransmitters. And I analyzed the hippocampus of different mammalian species that are used as animal models. And then from there, I just, out of the hippocampus, I moved to the rest of the brain. And I've been looking at the distribution of receptors. On the one hand, they help us find borders between areas, and this is one of the aims that I have, like to create a map of the brain. I'm concentrating on the macaque brain as an important model for research, but I also work with the human brain. And then the interesting thing about receptors is, or the receptors for classical neurotransmitters, is that they are the molecules in the brain that enable the transfer of information between nerve cells, right? So connectivity in the brain on the one side is axons and the fibers, and on the other side, once you get to the end of the axon and the synaptic button, you've got the neurotransmitters and the receptors. And we found that the distribution of receptors, it doesn't just show us borders between areas, but when you compare the expression levels of different kinds of receptors in the brain in different areas, this helps us find out things about the functional organization of these areas. So they're a really nice link between the structure and the function, right? I am an anatomist, and in anatomy we look for patterns. We have to name things so that you can describe them, so that you can talk to people and know that you're talking about the same structure. But it's not just about giving names, it's also about then trying to find out how this structure that you've identified could result in a certain brain function, okay? Because this is ultimately what we all want to know, how the brain works. Yes, so that's what I've been doing, like, in the past, and it's something that I will continue to do in the future. What I want to do in the future is try and collaborate with more intensely. I've already started doing so, but collaborate more with computer neuroscientists, because they can, I mean, when I look at histological sections, I can identify certain patterns, but there are patterns that I miss, and and that any other anatomist may also miss, and so on. But computers and deep learning procedures are helping us, not just speed up procedures, but also sometimes identify patterns that the human eye didn't detect. And what I think is really important if we want to advance our understanding of the brain is for anatomists and computer neuroscientists to work together, because only if we work together and in synergy and each one of us puts in their expertise, then AI sometimes produces beautiful images, but the computer neuroscientists just don't lack the training to interpret these images. And that's where then the anatomist comes in and says, well, knowing everything that I know about the brain, this could be this and that and whatever. And then this information can be fed back into the models and make them more complete. So this is what I think is most definitely going to be my future. I'm going to continue doing, hardcore anatomy because I enjoy doing it. But I'm definitely going to collaborate with people doing computer models and predictions and using AI procedures to help us understand this link between structure and function.
Thank you. Yeah, I think that's a really great answer. I think that integrating different disciplines in neuroscience, like you said, like computer neuroscience and anatomy, and making predictions is really interesting and very important. You also mentioned that your work spans across macaque and monkeys and humans. Do you see any challenges working with both within those models? Are there any benefits that you see by expanding your expertise into both?
Well, listen, our organization is called the Human Brain Mapping, right? And we humans are a little bit self-centered, and basically we want to understand how our brain works, right? Because we want to be able to find solutions when dysfunction basically results in a neurological or neuropsychiatric disorder. So that is the ultimate goal. It's the human brain. The problem is that there are certain experimental procedures that you can't do with the human brain, and for those things you need animal models. But then, of course, if you find something in an animal model, you've got to know how closely that model reflects what is happening in the human brain in order to be able to transfer the knowledge that you get from an animal model into the human world and basically ultimately help us find a solution for disease. So yes, we have to not just then understand the human brain, but we have to understand how our model brains are built so that we can, as I said, transfer the information between species.
Thank you. And across both models and throughout all of your work, what do you believe is your most significant contribution to the field? And of course, for anyone listening or reading the piece, we'll link those pieces as well.
Well, I think, this isn't a nice question. This is like asking a mother which one of your children is your favorite child. Yes.
Another way to ask could be, what do you think is the most, or is there a piece that's recent that you want to highlight or talk about more?
Well, okay, so there are two things that I like. I mean, one thing is all the studies that I've been doing on the distribution of receptor densities throughout the human brain and also throughout the macaque brain, and the information that has provided, the fact that it helps us identify areas from different functional systems and within a functional system, different hierarchical levels. So I think this is something that's really important, because, as I said, receptors, they are the structures that sort of like provide the link between the brain's structural segregation and its functional segregation. Okay, so that would be one thing. And then the other thing that I'm really proud of, and this first aspect, this is something that's basically my work. But in the latter years, I've been working on creating a 3D model of the macaque monkey brain, and I think that would be really helpful, given that functional imaging in the monkey brain is something that is now becoming much more frequent. So it will help the neuroscientific community, you know, at a global level. I think it will be a very helpful tool, but I'm also very proud of this because this is actually work that has been done by my PhD students and my postdocs, and it's nice to see their work, you know, out there and helping the community. So I'm proud of the Macaque Monkey Atlas for two reasons. It's also sort of like my baby, but it's my students' work, and they've done a great job. So yeah.
Great, thank you. And how do you envision the future of this field? Are there any developments that you're excited to see within the next 5 to 10 years?
Well, I'm looking forward to see what will happen with the computational neuroscience. I think it's a relatively young field, you know, in neuroscience, and I think that if computational neuroscientists and anatomists work together, it will help us advance. And I, you know, the caveat is that that we have to work together. I remember when functional imaging started to be, you know, the method to use, and people thought, fantastic, you know, we don't need anatomists anymore. We'll just put subjects into the scanner, we'll ask them to do a task, and then we'll understand how the brain works. And people that didn't know about anatomy, that didn't care about anatomy, they described the position of their activations, well, sometimes correctly and sometimes not so correctly. And you know, we still don't understand how the brain works, and the fact that sometimes, unfortunately, too often, things or activations were assigned to the wrong areas. It didn't help us advance the understanding of the brain. It made the waters like muddier than they were anyway. I think with all the data that's been collected, you know, in the past, and our ability to use deep learning procedures, it can help us speed up things. As I said, as long as, you know, when it comes to interpreting the results that you get, you really do ask somebody who knows about what the ground truth is, to, you know, not to create false statements and beliefs.
And what about the 7T MRI? Do you see this as the up and coming future for studying cortical structure in humans?
Yes, this could be very useful. I was actually involved in an article with some colleagues at the research center. They are physicists, and they were working with, you know, ultra high resolution functional MRI, and they could see layers in the primary motor cortex and the primary somatosensory cortex, and you know, it's easy enough to see layers in the primary visual cortex, because that's a part of the brain, of the human brain, that has such a distinct laminar structure that you don't even have to do a staining to be able to see it, right? And it has been shown in MR images, you know, in the past, but I'd never seen laminar patterns in the primary motor or somatosensory cortex in MR images before. So I'm really excited about that. And I think it will help because, again, doing studies in vivo, well, it's easier to get hold of volunteers. I don't know why people, they just don't want to, they don't mind going into the scanner for you, but they don't want to give you their brain.
Yeah. So talking about the 7T and some of the future of the field and knowing all of these advances in brain mapping, if you had the power to map anything in the brain, what would you do?
Oh, what part of the brain would I deal with?
Any part. What would you, if you had the power to do anything, and is there an area that you haven't explored or something that with the new tools you can use?
Right. Well, I would like to look at basically the limbic cortex, which is a whole region of the brain, which includes the hippocampus. The hippocampus was the topic of my PhD, so it's sort of like, it would be nice to get back to that in vivo at a really nice high resolution. That would be nice. I'd like, and then we've also got the cingulate gyrus, and it's the part of the brain that helps us, well, integrate our cognitive abilities and our emotional abilities. It's the part of the brain that lets us feel empathy. It's the part of the brain that doesn't work properly when you are depressed. This would be my favorite part of the brain. Yeah, the limbic system, definitely.
So speaking to when you were studying the hippocampus during your PhD, do you remember a specific moment where you knew, like, this is the topic I want to study and this is the field I want to be in?
A particular one.
Or is there anything that just really fascinated you that?
Well, yes. Okay. Yes. What fascinated me is that you can have one brain structure. So, for example, the hippocampus. And if you look at the hippocampus of one species with different histological procedures, it's fascinating the way these different images highlight different parts of the hippocampus. So if you look at a silver cell body staining, you basically just see like these two black C's that are interleaved. If you look at the myelin staining, you don't see those layers, but it highlights whatever the mossy fibers, the TIM staining super highlights the mossy fibers. The kainate receptors highlight the mossy fibers and the lucidum layer, just as the TIM staining does. But nobody else is interested in the lucidum layer and the mossy fibers, right? So I was fascinated by the fact that when you look at different molecular components of the brain, you highlight different parts of the same picture. It sort of like reminded me a bit of, I don't know if you know the image, Marilyn Monroe's photographs and Andy Warhol's paintings, the way he's used the different colors. Well, it's sort of like the same, but in the brain, okay? So I think it's fascinating that something that can provide so much information, these different stainings and so on, can at the same time also be so pretty.
Thank you. And your work, it's so interesting and it's important. And can you please describe your research interest and what you do to a lay audience?
Oh, okay. So, when I started doing my PhD, my mother asked me, what do you do? Now, what's the topic of your PhD? And I said, well, it's the distribution of receptors for classical neurotransmitters in the hippocampus of different mammalian species. And my mother said, it's the distribution of what for what in the what of different mammalian species. I said, okay, so right. What I analyze is the distribution of chemical substances that are used by brain cells to transfer information. Okay. And basically what you've got to imagine is that you've got, let's say, two cells in the brain, and one of them is dozing, and the other one receives input and says, hey, we've got to start work. So it will release this neurotransmitter, and this neurotransmitter will tell the cell that is dozing, it will say, hey, wake up, we've got to start working, because, you know, whatever the person wants to think. Or maybe, you know, the cell is working, and we want to tell it, look, you can stop working now. So again, another type of neurotransmitter will be released, and it will tell this cell, okay, slow down or stop altogether. Okay. And then it's this balance in the brain between the different cells telling each other to either speed up or slow down. This is what basically you know, lets the cells do a function. And I talked about this example with just two cells, but in the brain we've got millions and millions of cells, and they form networks. So they're groups of cells, and when a whole group of cells then works in synchrony, then, you know, a certain function is enabled, and depending on where those cells are located in the brain. And then you realize that you have seen something, you realize that what you have seen is a person, it is a person that you know, and this person is you know, moving away from you, for example. So all of these different kinds of input, it sounds very easy to say, you know, you've seen somebody that you know, but first your eyes have to receive this information, they have to pass it on to the brain, then that information has to be passed on to another part of the brain that tells you, oh, what you've seen is a person and not a building. And then that information is sent on to other parts of the brain that tell you, you know something, you know that person. And you know that person because that person is your sister or your friend, or, well, you actually don't know that person personally, but it's a famous actor and you've seen them in different films, right? So all of this kind of information gets integrated in the brain, and it's all basically thanks to the fact that that neurons talk to each other via these neurotransmitters that either tell them to speed up their level of activity or slow it down.
Thank you so much. You gave us some of your highlights from your work and also told us about why you're passionate about the field. We're going to shift the focus a little bit more to your career now. What are some of the biggest obstacles you've had to overcome in your career?
I don't like politics. I think that was the biggest obstacle that I had to overcome. I do science because I want to understand how the brain works, right? And that's okay, if you know, if you want to stay at a certain level, that is super important in the brain. I also wanted to, you know, to be able to train future scientists, PhD students. If you want to recruit PhD students, you have to teach at university. If you want to teach at university, you do sort of have to follow a certain career. If you want to be able to officially supervise these students, you have to have a certain level. So it means then you have to do these steps. You can't just be a PhD student all your life or a postdoc all your life. If you want to do research, you have to get funding. So you can't just be sit in your office looking down the microscope all day. Yes, you have to do this, and it is something that I accepted very late in life. But then I did it, and it actually doesn't hurt. I don't think I don't think that it has made me a better scientist. I don't think it has made me a better person, the fact that I'm now a professor. But while it helps because I can supervise PhD students, and my students are happy with me up to now anyway, so we'll see.
So speaking of PhD students and future scientists, is there any advice that you would like to give early career OHBMers?
You've got to be passionate about what you do. You really have to love it, because it involves a lot of work and unfortunately a lot of frustrating work. You do research, you have a question, and you think that if you do this and this experiment or this and this analysis, you'll get the answer to your question. If you're lucky, you do get at least a partial answer to your question. If you're not lucky, it doesn't answer your question, but it opens other questions, right? And I said, if you're not lucky, it doesn't answer your question, because traditionally in science, when you publish your results, journals have always wanted you to publish like positive results. If you're looking, say you're comparing a certain brain region of a healthy control and of a patient with a certain disease, journals want you to report that there's a significant difference in whatever, right? But if your results say that for whatever it was that you were looking at, there is no significant result, well, that's also a finding, only that is always more difficult to publish than a significant finding. So that can be frustrating, sending your articles off, getting rejections because your topic of research isn't sexy enough, or it can be very frustrating. Writing grant proposals that are, on topics that are actually super interesting, but them not getting funded because there's just, not enough money anymore, and there are too many people asking. Don't get frustrated. As long as you're, passionate about what you're doing, it's okay. And you just have to keep on fighting. And it's also okay to talk about being frustrated, right? I also went through phases when I was frustrated, and it helps to tell your supervisor, or send me an email and say, I'm frustrated because what would you do, or whatever. So talk about it with your supervisor. This is something that's very important. And if for whatever reason you can't talk to your supervisor, look for somebody else that you can talk to and ask for counsel, because it can be very frustrating, right? But then when an article gets accepted, it's like, whee, it's nice. When you get invited to give a talk, it's nice. And this is one thing that is important for the early career students, and what I would also tell the more experienced postdocs is, to also give their students the chance sometimes to present their own stuff. There's this balance between postdocs getting invited to give talks because their work is, more known, but because they've been in the field longer and they've published more, basically. But sometimes also give your students the chance to be the ones to present their own work, because that it's a nice feeling, and it helps sometimes to overcome this bit of when one is frustrated.
Thank you so much. And we are on to our last question. And this is what I know everyone is excited to hear. Can you give us a little sneak peek to your upcoming OHBM talk?
Well, I did have to write the abstract the other day, so that's why I know. Well, no, I don't quite know. What I want to talk about is how receptors have helped us in the past understand the structural segregation in the brain and the insights that they have, provided also concerning these organizational principles in the brain. But I also then want to move towards this, the fact that when we've got the same receptor type that has been analyzed in post-mortem studies, this is what I've been doing all the time, it is comparable to what we can find in vivo with PET studies. And here's where I also think, computational neuroscientists will help. The ligands that are available post-mortem to analyze different types of receptors, there are many more available for post-mortem studies than for in vivo studies. And given the fact that we know for those cases, for those receptor types where we can analyze them post-mortem and in vivo, that post-mortem is, a good prediction of what happens in vivo, I think that then we will be able to, create models and transfer all of this post-mortem data into in vivo models. And I have a collaboration with Yasa Ituria Medina at the MNI, and he's creating beautiful personalized models. So this will help us, advance the understanding of the changes of receptor level that happen in patients. So this is a bit the overview. It's what I consider the past of receptors, the post-mortem, and then the fact that they can be used for in vivo studies and personalized brain models.
Great. Thank you so much for your time. And I really enjoyed talking with you, and congratulations again on your upcoming keynote. And we're all looking forward to hearing from you.
Well, it was very nice talking to you as well. And yes, looking forward to see it.
Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.